Hello dear friends, once again it's very hearty and warm welcome. Dear friends, in our today's lecture we are going to study about post-fertilization changes in angiosperms. All of you are known in our previous lecture, we people have studied about the mechanism of double fertilization. And now here we have to study about post-fertilization changes. Actually, the title itself indicates what we people have to study in this topic. Post stands for after. Post-fertilization changes. It means after the process of fertilization, the ovule undergoes some noticeable changes so that it gets converted into seed. In the same manner, the ovary wall also undergoes some noticeable changes to form fruit. And hence, we can say the formation of seed from ovule or development of seed and the formation of fruit from ovary is nothing but the post-fertilization changes. Let me tell you that soon after the process of double fertilization in angiosperms, the flower begins to lose its shine. Soon after the process of double fertilization ची प्रोसेस झाल्यानंतर in that angiospermic plant flower it begins to lose its shine. The different floral parts, say the petals, stamens, even the stigma and style breaks off, wither away, dries away. But sometimes the sepals persist up to the formation of fruit. It means in some flowers, in some plants, the sepals persist up to the formation of fruit, and in some plants, the sepals also wither away, dries off and fall down. Say, all of you are known in case of tomato, in case of brinjal, the sepals are seen, sepals persist up to the formation of fruit also. Now, here we people have to study the two major post-fertilization changes. First, that is formation of seed, and second, formation of fruit. So let us start first of all the study of formation of seed. Formation of seed. Just as I told you that after the process of double fertilization, ovule undergoes some noticeable changes and hence it gets converted into the seed. And that's why in previous lectures I told you the ovule is regarded as forerunner of seed. Now, before starting the discussion of this topic, here I am going to draw a diagram of anatropous ovule. I am going to show the diagram of anatropous ovule in which already the zygote, the embryo sac is there with zygote and triploid pair. Please look at the diagram. Consider here is supposed to be the ovary wall. Just consider here is supposed to be the ovary wall. Now, in this ovary, consider here is only single anatropous ovule for our convenience. We all are known anatropous, that is inverted ovule, externally surrounded, protected by means of mostly two protective coverings, that is integuments. So here are two integuments, outer and inner integuments. Here is micropyle chalaza, we all are known. Now I am going to show embryo sac. Here is the embryo sac. You know that embryo sac now contains two main components. First one is zygote. With the help of pink color, I have shown the zygote. Here is the zygote, diploid zygote. It is the product of syngamy or first fertilization. And here I am going to show with the help of blue chalk triploid pane. Consider here is a triploid pane. You know that triploid primary endosperm nucleus. Okay, and it is the product of second fertilization or triple fusion. Now with the help of yellow chalk I have shown the embryo sac. Embryo sac. Of course, now embryo sac contain zygote towards the micropylar end and a triploid pane. This embryo sac is surrounded by means of a thin layer of nucellar tissue, nucellus. This nucellus may persist up to the formation of seed also, and this thin layer of nucellus, if present in seed, then it is known as perisperm. Perisperm. So here I have shown the ovary in which anatropous, fertilized anatropous ovule is present. In this fertilized anatropous ovule, here we have shown embryo sac enclosing diploid zygote towards the micropylar end and centrally placed triploid pane. Now I said that during the formation of seed, this ovule undergoes certain noticeable changes, and due to which it gets converted into the seed. Three major changes takes place during the formation of seed from ovule. Three major changes takes place. Let us see one by one each. The first change is formation of endosperm. Formation of endosperm. Endosperm is nothing but the food-laden tissue, or we can say endosperm is nothing but it is the nutritive tissue. It is the nutritive tissue, and this endosperm provides nourishment, provides nutrition to the developing embryo, as well as it provides nourishment during the process of seed germination for the young seedling, also. So first of all we have to remember that endosperm is nothing but it is the food-laden tissue or nutritive tissue, and it provides nutrition, nourishment for the developing embryo, as well as it provides nourishment during the process of seed germination and formation of young seedling. This endosperm in angiosperm develops from triploid pane. In angiosperm, endosperm, it develops from triploid pane, and it develops only after the process of double fertilization. Try to understand. एंजिओस्पर्म्स मध्ये हा जो एंडोस्पर्म आहे, एंडोस्पर्मिक टिश्यू आहे, न्यूट्रिटिव्ह टिश्यू आहे, हा ट्रिप्लॉइड पेन पासून विकसित होत असतो आणि तो डबल फर्टिलायझेशन झाल्यानंतरच तयार होतो, आणि म्हणून इन एंजिओस्पर्म दिस एंडोस्पर्म इज ट्रिप्लॉइड इन नेचर. इट इज ट्रिप्लॉइड इन नेचर and post-fertilization tissue. It is regarded as post-fertilization tissue. While in gymnosperms, the endosperm is haploid in nature and it is pre-fertilization tissue. Means in gymnosperm, endosperm develops before the process of fertilization, but here in angiosperm, endosperm, it develops only after the process of double fertilization, and as it develops from triploid pain, hence it is also, endospermic tissue is also triploid in nature, and it is known as the post-fertilization tissue. Now, on the basis of mode of development of endosperm, there are three types. Let me tell you. हा एंडोस्पर्म कशा प्रकारे तयार होतो याच्या आधारावर एंडोस्पर्मचे तीन प्रकार पाडले जातात. सो लेट अस टॉक वन बाय वन ईच. फर्स्ट वन is known as nuclear endosperm. Nuclear endosperm. Listen. This type of endosperm is frequently seen in angiospermic plants. Let me tell you, in about 161 angiospermic families, this type of endosperm is seen, that is nuclear endosperm. Let us talk about its development. For that purpose, take out this embryo sac. Consider this embryo sac. Temporarily, just consider the triploid pain. Triploid primary endosperm nucleus, and you know it is surrounded by cytoplasm. तात्पुरतं आपण या ठिकाणी झायगोट कन्सिडर केलेला नाहीये, कारण आपल्याला माहिती आहे की एंडोस्पर्म हा ट्रिप्लॉइड पेन पासून डेव्हलप होणार आहे, म्हणून आपण फक्त या ठिकाणी एम्ब्रियो सॅक आणि त्या एम्ब्रियो सॅक मध्ये असलेला ट्रिप्लॉइड पेन कन्सिडर केलाय. नाउ ड्यूरिंग द डेव्हलपमेंट ऑफ न्यूक्लियर एंडोस्पर्म, दिस ट्रिप्लॉइड पेन undergoes repeated mitotic and free nuclear division. This triploid pain undergoes repeated mitotic free nuclear divisions. It means here it occurs the free mitotic nuclear divisions only, karyokinesis only, and karyokinesis is not immediately followed by cytokinesis. या ठिकाणी न्यूक्लियर एंडोस्पर्म तयार होत असताना हा जो ट्रिप्लॉइड प्रायमरी एंडोस्पर्म न्यूक्लियस आहे त्याचं एका पाठोपाठ एक सातत्याने फ्री मायटोटिक डिव्हिजन्स होतात. मायटोसिसच्या पद्धतीने त्या न्यूक्लियसचं विभाजन होतं. पर्यायाने देयर आर फॉर्मेशन ऑफ मेनी फ्री ट्रिप्लॉइड न्यूक्लियाय, मेनी फ्री ट्रिप्लॉइड न्यूक्लियाय. अँड ॲज आय सेड, आता जसं मी म्हटलं कि nuclear division is not followed by cytoplasmic division, karyokinesis is not followed by cytoplasmic division, and that's why there are formation of number of triploid nuclei in the central cell of embryo sac. Central cell of embryo sac. These all are the triploid nuclei. It is followed by the formation of a central vacuole, and this central vacuole it enlarges. Central vacuole it enlarges in its size. Due to this enlarged central vacuole, the triploid nuclei, these triploid nuclei get pushed along the periphery. Consider here is the central vacuole, large central vacuole, and due to this large central vacuole, all these triploid nuclei get pushed along the periphery. Hence we can say that triploid nuclei are placed in between the wall of embryo sac and central vacuole. You know, here is the wall of embryo sac and here is the central vacuole. Now, initially, that free nuclei remains as it is, and it acts as the endosperm. As here, endosperm is represented by many free triploid nuclei. Therefore, it may be known as the nuclear endosperm. But later on, in later stage of development, it starts the formation of cell walls centripetally, cell walls centripetally, that is from periphery towards the center. काही ठिकाणी हळूहळू नंतर पुढच्या अवस्थेमध्ये सेल वॉल फॉर्मेशन सुरू होतं, सेल्स तयार व्हायला लागतात फ्रॉम पेरिफेरी टू द वी कॅन से सेंटर. In coconut, the peripheral endosperm is cellular and the internal is nuclear. कोकोनट मध्ये जो पेरिफेरल एंडोस्पर्म आहे, कडेचा जो एंडोस्पर्म आहे तो सेल्युलर असतो, म्हणजे सेल्सच्या स्वरूपात असतो आणि आतल्या बाजूने मात्र एंडोस्पर्म इज इन द फॉर्म ऑफ फ्री ट्रिप्लॉइड न्यूक्लियाय. Anyway, so here I told you about the development of nuclear endosperm. A typical example is of wheat. Now come to the second type. Second type of endosperm is known as cellular endosperm. Cellular endosperm. Very simple. Cellular endosperm, it is seen in Datura, balsam, etc. In cellular endosperm, the triploid pane divides mitotically, and this nuclear division is immediately followed by cytoplasmic division. Let me show in the diagram. Here is the triploid pane, here is the triploid pane, and this triploid pane undergoes karyokinesis to form two daughter nuclei, and these two daughter nuclei, after the formation of two daughter nuclei, immediately there is a formation of cell wall in between two daughter nuclei. Now this first cytoplasmic division may be either transverse, may be vertical, may be oblique. It means here after every karyokinesis, after every nuclear division, it occurs cytokinesis, that is division of cytoplasm. या ठिकाणी प्रत्येक वेळेला न्यूक्लियसचं डिव्हिजन झाल्यानंतर, कॅरिओकायनेसिस झाल्यानंतर ऑटोपार्ट सायटोकायनेसिस होतं, सायटोप्लाझमचं डिव्हिजन होतं, डॉटर न्यूक्लियाच्या मध्ये सेल वॉल तयार होते, and hence we can say it leads to the formation of mass of many triploid cells. It leads to the formation of mass of many triploid cells, and such mass of triploid cells is nothing but the cellular endosperm. Cellular endosperm. Here we have considered second type, example Datura. Now third type is known as halobial endosperm. Halobial endosperm. Halobial endosperm is seen in angiosperms belonging to the family Halobiaceae. Example Asphodelus. Now let me explain you about the halobial endosperm. In this type, actually the development occurs just as intermediate between the cellular and nuclear type of endosperm. Listen. Halobial endosperm is considered as intermediate type of endosperm of cellular and nuclear, because it shows some features, some characters like nuclear endosperm and some features like cellular endosperm. Let me show and let me explain you. Consider once again, here is embryo sac and triploid pane. Initially, the triploid pane, it divides mitotically to form two daughter nuclei. Means it occurs the karyokinesis, and this karyokinesis, that is division of nucleus, is immediately followed by cytokinesis that is cell wall formation, but the cell wall is formed in such a way that the embryo sac, central cell of embryo sac, get divided into two unequal cells. One is very small towards the chalaza and one is very large. So we can say here is the chalazal cell and here is a micropylar cell. It means it occurs in karyokinesis followed by cytokinesis, and now out of these two cells, this chalazal cell, nucleus of chalazal cell, either divides or remains as it is. It divides or remains as it is. Suppose, let me show, here it occurs the karyokinesis leading to the formation of many daughter nuclei, many triploid daughter nuclei, while this larger micropylar cell shows karyokinesis followed by cytokinesis. It means it occurs the division of nucleus immediately followed by the division of cytoplasm, division of nucleus followed by division of cytoplasm, leading to the formation of mass of many triploid cells. Mass of many triploid cells. It means here in this type of endosperm, some part represent the cellular endosperm and some part represents nuclear endosperm, and that's why I said it is supposed to be the intermediate type of endosperm to the nuclear and cellular. One more thing, mosaic endosperm is seen. Mosaic in maize, Z maize, mosaic endosperm is seen. Example, Z maize. In Z maize, the endosperm is not homogeneous. It is not homogeneous, but the endosperm, it appears in the form of patches of red and white color, and this mosaic endosperm was discovered by a scientist Weber. It is one more type. Anyway, so here just now we have completed the study of first change in ovule towards the formation of seed, that is formation of endosperm. Now please look in this diagram. या ठिकाणी या डायग्राम मध्ये आपल्याला एम्ब्रियो सॅक दिसतोय, ट्रिप्लॉइड पेन दिसतोय. मग अर्थातच या ठिकाणी या ट्रिप्लॉइड पेन पासून एंडोस्पर्म तयार झाल्यानंतर या ठिकाणी पूर्णतः हा एंडोस्पर्म प्रेझेंट आहे. एंडोस्पर्म प्रेझेंट आहे. I told you that endosperm is a nutritive tissue. It provides nourishment for the developing embryo. Now we have to study the development of embryo, process of development of embryo. But the seeds in which endosperm is not completely utilized by embryo, and thus if present in resultant seed, then it is known as endospermic seed. Say for example wheat, and the seeds in which the endosperm is completely utilized by developing embryo, and thus there is no endosperm in the seed, such seeds are called as the non-endospermic seed. Say for example pea, bean, etc. नावच सांगून जातो. एंडोस्पर्मिक सीड याचा अर्थ ज्या सीड मध्ये एंडोस्पर्म शिल्लक आहे अशा सीड्सला एंडोस्पर्मिक सीड्स म्हणतात किंवा अल्ब्युमिनोस सीड्स पण म्हणतात. आणि ज्या सीड्स मध्ये एंडोस्पर्म हा पूर्णतः युटिलाइज केलाय आणि म्हणून आता सीड मध्ये एंडोस्पर्म शिल्लक नाही अशा सीड्सला म्हटलं जातं नॉन एंडोस्पर्मिक किंवा नॉन अल्ब्युमिनोस सीड्स. ओके, फॉर एक्झाम्पल पी, बीन. Now we have to study about the second change, that is formation of embryo from zygote. Let us study the second step in the formation of seed, that is development of embryo. Friends, just now we have completed the study of first major change taking place during the formation of seed, that is formation of endosperm from triploid pair. And now let us talk about the development of embryo. First of all, let me define what is embryo. Actually, in plants, the embryo is defined as well organized young miniature plant. In plants, the embryo is defined as well organized young miniature plant. Young miniature plant. Now, why this is so? To understand its meaning, let me first of all explain you briefly about the structure of dicot embryo and also about the structure of monocot embryo. Actually, we people have studied about the structure of dicot and monocot embryo in our 11th class. But then and then, let us see once again. Consider, here is supposed to be the dicot embryo. Now, the dicot embryo, it consists of two main components such as embryonal axis. Consider, here I am going to show the embryonal axis. Embryonal axis. Now, this terminal end of embryonal axis represents the radicle. The other opposite end of embryonal axis represents plumule. Now, in between radicle and plumule, a region, a point where the cotyledons are attached to the embryonal axis is known as first node. Let me explain you. Suppose these are the cotyledons. Just consider these are the cotyledons. We know that there are two cotyledons because it is dicot embryo. Name itself indicates it is dicot embryo. So these are the two cotyledons. And I say that the point or the region of embryonal axis at which the cotyledons are connected to the embryonal axis is known as first node. So consider this is supposed to be the first node. Now the region or the part of embryonal axis between first node and radicle is known as hypocotyle. Hypocotyle. Please look at the diagram. With the help of pink color I am going to show. It is hypocotyle. The region of embryonal axis in between the radicle and first node is known as hypocotyle, and the region of embryonal axis between plumule and first node is known as epicotyle. Epicotyle. Here with the help of blue chalk I have shown the epicotyle. Okay, and these two yellow colored structures are nothing but the cotyledons. Now you just consider in this dicot embryo, these two cotyledons represent the future leaves. Radicle represents the root or root system, while the plumule it represents the shoot, or we can say shoot system in plant body, and that's why I said actually the embryo is defined as well organized but young miniature plant. So here briefly I explained you the main features of dicot embryo. Main features of dicot embryo. Now here I am going to draw the diagram of monocot embryo. The monocot embryo, name itself indicates there is single cotyledon. Let me draw. Here in monocot embryo there is a single cotyledon. It is present at one side, and the embryonal axis, here is the embryonal axis, its one end as usual represents radical, another end represents plumule, and one more thing, the radical and plumule covered by means of protective cap-like structures. Protective cap-like structure. A protective cap over the radical is known as coelurhiza. Coelurhiza. While the protective cap over plumule is known as coeloptile. Coeloptile. Anyway, so here first of all, before starting the study of actual development of embryo in dicot and monocot, I have explained you briefly the structure of dicot and monocot embryo. Now let us start the study of this topic. Here in bracket already I have written the development of embryo is also known as embryogeny or embryogenesis. एम्ब्रियो तयार होण्याच्या प्रक्रियेला एम्ब्रियोजेनी किंवा एम्ब्रियोजेनेसिस असं म्हणतात. या ठिकाणी मी आत्ताच तुम्हाला डायकॉट एम्ब्रियो, टिपिकल डायकॉट एम्ब्रियोची सर्वसाधारण रचना आणि टिपिकल मोनोकॉट एम्ब्रियोची सर्वसाधारण रचना या ठिकाणी काढून दाखवलेली आहे. आणि आता प्रत्यक्ष आपल्याला या ठिकाणी हे एम्ब्रियो कशा प्रकारे तयार होतात हे शिकायचं आहे. ऑल ऑफ यू आर नोन the zygote leads to the formation of embryo. Okay, now please look at the blackboard. Once again, here I am going to draw the anatropous ovule. I am going to draw the anatropous ovule. Here is the anatropous ovule, because we know in majority of the angiospermic plants the ovule is inverted anatropous type. So here I have shown the integuments, micropyle. As usual, we know I am going to show here the embryo sac, and already we have studied in this embryo sac. Now the triploid pane is converted into the endosperm. So this is food laden tissue, nutritive tissue, that is endosperm, and we know that here at the micropylar end presence of zygote, diploid zygote, isn't it? Now we know that this diploid zygote, it gives rise to the embryo, and for that purpose, to understand the actual mechanism of development of embryo, just take out this zygote outside this embryo sac, outside this ovule. Actually, the development of endosperm and development of embryo in angiosperm takes place simultaneously. Try to understand. Angiosperms मध्ये endosperm ची डेव्हलपमेंट आणि या झायगोट पासून एम्ब्रियोची डेव्हलपमेंट ही सायमल्टेनियसली घडून येत असते. आता आपल्या सोयीसाठी, for our convenience, first of all we have studied the development of endosperm, and now let us talk the development of embryo. So let me draw here the zygote. Here is a diploid zygote. It is a single cell structure. Zygote. Initially we people have to study the development of dicot embryo. Development of dicot embryo. Typically, during the development of dicotyledonous embryo, dicot embryo तयार होत असताना, initially the zygote, it slightly swells in its size and secretes its own cellulosic cell wall. I said that during the development of typical dicotyledonous embryo, डायकॉट एम्ब्रियोची डेव्हलपमेंट विकास होत असताना सुरुवातीला झायगोट इट स्लाइटली एनलार्जेस इन इट्स साइज, झायगोट स्लाइटली एनलार्ज होतो and it secretes its own cellulosic cell wall around itself. Now zygote it divides mitotically and unequally by transverse division. I said that zygote it divides mitotically, that is by mitosis, unequally and transversely to form two cells, two unequal cells. One is smaller cell and another larger cell. One is smaller and another larger cell. The larger cell which lies towards the micropyle, you must have proper understanding. This larger cell which lies towards the micropyle is known as basal cell. It is known as basal cell, or it may be known as suspensor cell. Basal cell or suspensor cell. While another smaller cell which lies towards the center of embryo sac, दुसरी जी स्मॉल सेल आहे की जी एम्ब्रियो सॅकच्या सेंटर कडे आहे, that smaller terminal cell is known as embryonal cell. Terminal or embryonal cell. Now this basal cell, suspensor cell divides transversely, while the embryonal cell divides vertically, of course by mitosis. Because you must know, during the development of embryo, whatever cell division take place, that all are mitotic. अर्थात एम्ब्रियोची डेव्हलपमेंट होत असताना जेवढ्याही प्रकारचे पेशी विभाजन, सेल डिव्हिजन होणार आहे ते सगळे मायटोसिस या पद्धतीने होणार आहे. So I said that now the suspensor cell divides transversely and terminal cell, embryonal cell divides vertically. Let me show. Of course, leading to the formation of four cells. Let me show here in diagram. So I said the suspensor cell it divides transversely to form two cells, and embryonal cell divides vertically again to form two cells. Actually, this stage is known as quadrant stage. It is known as the quadrant stage. Okay, because here are four cells. The two cells formed by the division of suspensor cell, of which the lower cell, that is towards the micropyle, सस्पेन्सर सेलच्या डिव्हिजन मधून या ज्या दोन सेल्स तयार झालेल्या आहेत त्यापैकी मायक्रोपाईल कडे असणारी अर्थात जी लोअर मोस्ट सेल आहे, that lowermost cell it enlarges in its size. The lowermost cell enlarges in its size. This enlarged cell is known as hostorium. It is known as hostorium, or it may be called as vesicular cell, because it appears just like a vesicular cell. While the another cell formed by the division of suspensor cell, it redivides mitotically and transversely to form a row of 6 to 8 cells, placed one above the other. Please look at the diagram. मी आता असं म्हटलं की या ठिकाणी सस्पेन्सर सेलच्या डिव्हिजन मधून ज्या दोन सेल्स तयार झालेल्या आहेत त्यापैकी जी लोअर मोस्ट सेल आहे ती एनलार्ज होते आणि या एनलार्ज लोअर मोस्ट सेलला हॉस्टोरियम किंवा वेसिक्युलर सेल म्हणतात आणि दुसरी जी सेल आहे तिचं मायटोसिसच्या पद्धतीने विभाजन होतं अँड अल्टिमेटली देयर इज अ फॉर्मेशन ऑफ अ रो cord of 6 to 8 cells. That cells are placed one above the other in the form of row, leading to the formation of a cord-like structure known as suspensor. This structure is known as suspensor. Now in this suspensor, the terminal cell which is nearer to the embryonal cell, या ठिकाणी ही जी टोकाची सेल आहे की जी एम्ब्रियोनल सेल्सच्या जवळ आहे, this cell is known as hypophysis. It is known as hypophysis. Now the two cells formed by the division of embryonal cell or terminal cell again divides vertically and it is followed by transversely. Just consider, या ज्या दोन सेल्स तयार झाल्या आहेत एम्ब्रियोनल सेलच्या विभाजनातून, त्यांचं आधी व्हर्टिकल डिव्हिजन होतं, अर्थात देर आर फॉर्मेशन ऑफ ग्रुप ऑफ 4 cells. You must have understanding. In this manner I am going to show. 2 cells are towards front side, 2 cells at the back side, because these 2 cells get divided vertically. Now there is a formation of group of 4 cells. Is that clear? Actually this embryonal cell divides, redivides and gives rise to the embryo proper. That's why actually this cell is said to be embryonal cell. Now this group of 4 cells is known as 4-celled stage of embryo. It is 4-celled stage of embryo, and these 4 cells further divide horizontally, further divides transversely to form 8 cells, to form group of 8 cells. You know, it is known as the octant stage of embryo. Octa stands for the 8. So now here is a formation of, we can say, octant embryo or octant stage of embryo. Octant stage of embryo. Later on, the cells of octant stage of embryo divides, redivides rapidly along many different planes to form a mass of cells, globular mass of cells. Globular mass of cells. Listen. The cells of octant stage of embryo divides, redivide mitotically and form a globular mass of cells. Globular, that is rounded mass of cells. It is known as globular embryo because it appears just like a globose, rounded. That is globular embryo. Globular embryo. Remember, the suspensor still persists. सस्पेन्सर आहे, या ठिकाणी मी काढला नाही याचा अर्थ असा होत नाही की सस्पेन्सर नाहीये. सस्पेन्सर आहे. बेसिकली द मेन रोल ऑफ सस्पेन्सर इज टू ब्रिंग the developing embryo close to the endosperm. Means in other words we can say the suspensor it pushes the developing embryo towards the endosperm to get to obtain the nourishment, nutrition. Anyway, so I said that here is a formation of globular embryo. Now this globular embryo slowly gets converted into heart-shaped embryo. Heart-shaped embryo. Let me show. Heart-shaped embryo. This is because of formation of radical, hypocotyle and less distinct cotyledons. less distinct cotyledons. Please look here. I am going to show this is radical, then its lower part hypocotyle, and these are less distinct cotyledons. Due to the formation of less distinct cotyledon, the globular embryo gets converted into heart-shaped or chordate embryo. Heart-shaped or chordate embryo. I will say chordate or heart-shaped embryo. Heart-shaped embryo. Now, in further development, the plumule, epicotyle and cotyledons become very much distinct, and the embryo is curved very much. Let me show. The embryo is curved very much. Here I am going to show the radical, hypocotyle. These are two distinct cotyledons, and this middle part is nothing but the plumule along with the epicotyle. So, such I will say curved embryo is also known as horseshoe shaped embryo. Horseshoe shaped embryo. And in this horseshoe shaped embryo, the distinct parts of dicot embryo are formed, such as with radical, hypocotyle, plumule, epicotyle and two cotyledons. In this manner, there is a formation of dicot embryo. Of course, after the complete formation of embryo, here the suspensor it degenerates, and now that embryo it stops its growth temporarily. It becomes dormant, inactive. It becomes active later on during the germination of seed. अर्थात इथपर्यंत वाढ झाल्यानंतर त्या ठिकाणी हा जो सस्पेन्सर आहे हा डिजेनरेट होतो आणि एम्ब्रियो हा डॉर्मंट स्थितीत, इनएक्टिव्ह स्थितीत, अक्रियाशील अवस्थेत सीड मध्ये तसाच राहतो आणि ज्या वेळेला सीड जर्मिनेशन होतं, बीजांकुरण होतं, त्यावेळेला that embryo again resumes its metabolic activity and it starts its further growth for the development of seedling. Here we have discussed about the development of dicot embryo, but let us talk little bit about the development of monocot embryo. I already told you that during the development of monocot embryo, the initial developmental process is same just like the dicot embryo. अर्थात मोनोकॉट एम्ब्रियोची डेव्हलपमेंट होत असताना सुद्धा सुरुवातीची जी अवस्था आहे, सुरुवातीच्या डेव्हलपमेंटल ज्या स्टेजेस आहेत त्या डायकॉट एम्ब्रियो प्रमाणेच असतात, बट इन लॅटर स्टेजेस it occurs only the development of single cotyledon, and such single thick and shield shaped cotyledon in monocot plant is known as scutellum. Of course, the plumule and radical are along the lateral side of scutellum, along the lateral side of cotyledon, and that plumule and radical remain covered by means of protective coverings such as coleoptile and coelorhiza respectively. In this manner, here we have studied about the development of embryo. All of you are known, we people studying about the development of seed from ovule, and already told you that during the development of seed from ovule it occurs three major changes. First, the development of endosperm from triploid pen. Second, development of embryo from zygote, just now we have completed. And third change, that is the formation of seed coats. Formation of seed coats. We know that the integuments, that is outer protective coverings of the ovule, integuments, आपण ओव्ह्यूल शिकत असताना पाहिलेले आहे. The integuments are mostly two in number in bitegmic ovule. Now the outer integument, it becomes hard, thick, leathery and gives rise to the outer seed coat known as testa. Outer seed coat known as testa. And if inner integument persists, it gives rise to the inner seed coat known as tegmen. So the outer seed coat testa and inner tegmen are formed from integuments. The micropyle, small terminal opening, isn't it? This micropyle persists as a very small slit, very small opening on the seed coat, and it is necessary for the entry of water as well as oxygen during the soaking of seed, and it is necessary for the germination of seed. याचा अर्थ असा होतो की या ठिकाणी हे जे मायक्रोपाईल आहे ते जवळपास बंद होतं, पण तरी सुद्धा अतिशय सूक्ष्म स्वरूपात मायक्रोपाईल एका स्लिटच्या स्वरूपात त्या ठिकाणी सीड कोटवर असतं. आणि ते कशासाठी गरजेचं आहे? इट इज नेसेसरी फॉर द एंट्री ऑफ वॉटर अँड ऑक्सिजन ड्युरिंग द सोकिंग ऑफ सीड, and that water and oxygen is necessary for the germination of seed, for the development of embryo into the new seedling. So here we have completed the study of first post fertilization change, that is formation of seed, development of seed from ovule. Okay, now the second post fertilization change is formation of fruit. That definitely we shall study immediately in our next lecture. Till then, thank you. Let us stop here. Bye.